So, hi everyone, welcome to Biodynamics Days 2025. It's our pleasure to have today Human Rezaei from INRAE in Paris, France, and he will talk about prion pathologies, intrinsic dynamics, and deterministic diversification drive, a new model of prion replication and dissemination.
So thank you very much. I hope that you hear me correctly. Everything's okay. Perfect. So first of all, thank you to Benjamin to invite me to make a talk here, and also thank you to all the organizers. So thank you. So as Benjamin said, today I'm gonna talk about prion pathologies and how the dynamics of the prion assemblies contribute. The study of the dynamic assemblies contribute to establish a new replication model and disseminations. So before start, I will make some introduction. Something very important. I'm not mathematicians. I am a biophysicist. So by advance, I apologize by the fact that I will not be able to address the mathematical aspect of this project, and Mathieu, that he will start immediately after me, gonna go much more deep in the mathematical aspect of the subject. So, few words about the prion pathology. So, prion pathology is a neurodegenerative disease. It induced motor and sensitive dysfunctions. It's always associated with neural loss, spongiosis, and astrocytosis. What does it mean, neural loss? It means that you lose your neurons and you have a vacuole in your head. Spongiosis, it means that it's like a spongios because you lose your neurons and you have vacuole in your head. Astrocytosis, you have a lot of proliferations of cells that are not able to are involved to clean the place. And this pathology is always associated with accumulation of misfolded and aggregated PRP. PRP is a very small protein. I will come to this point a little bit later. So usually you have a long asymptomatic incubation period. In human, it means that, for example, you will be infected by prions today, but you're gonna die in 20 years, 15 and 20 years. So you have a very, very long incubation period. The issue is fatal. I mean that when you are infected, no hope, you're gonna die, whatever. Without any conventional immune and inflammatory reactions, it means that when you have infected, you can go to your doctor, make a lot of analysis, but nobody can tell you that you have prion disease. And yes, of course, absence of surgical symptom. And this pathology is transmissible. It means that if I take your brain, your blood, and I put it in another individual, this individual will develop also the pathology. And this pathology is always associated with the conformational chain of PRPC, that's a good proteins, into a bad folded proteins that called PRPSC and aggregated one. And this is the heart of our studies. So the prion pathology has triple determinism. It can be sporadic. So in Europe, it's 1 or 2 cases per year per millions of people. For example, in France, we have almost 100, 150 persons per years. It could be infectious. As I told you, I take the brain of someone that's infected by prions. I put it in the body of another one, and he will develop the pathology. And typically, you have the crew determinist that people catch this pathology through by cannibalism. You have the variant of Creutzfeldt-Jakob, VCGD. That's the consequence of the mad cow disease. And iatrogenic, it's in France we had more than 800 people, child in late 70s that received growth hormone that was purified from human brain, and they develop this pathology, and now they almost all passed. And you have also the genetic form that this genetic form is different type of mutation in PRP that induce what we call familial CGD, Grassmann-Strauss-Schinkel GSS pathology, or fatal familial insomnia. This last is quite interesting because they don't die because they lose their neurons, but some disorder in their brain make that they cannot sleep, and they die by tiredness. In fact, they cannot sleep, and so they died finally. The evolution of the pathology, a few words, but at this point is quite important also for the next part of the talk, is that when you inoculate, you infect, for example, an animal for experiments, you have aggregations and accumulation of prion PRPSC, the bad form, in the brain during, let's say, the first phase of the pathology. And then you reach a plateau and nothing changed. And then from a mechanism that is not yet clear, you start to have neural disorders, and the neural disorders and neural death arrived late in the late stage of the pathology. So let's say that the research in prions start, let's say, more than 60 years ago. And since 60 years ago, we had only two models of prion replications in terms of mechanism, in terms of kinetics. The first models come from Griffiths, that was a mathematician, and he only observed the epidemic of scrappy disease. That's a kind of prion disease also in sheep. He observed and he proposed a very simple mechanism which not involved at all any conventional pathogens, not virus, not bacteria, not parasites. And he said, okay, you can have two kind of proteins, A and B, and you have the B that come and use the conformational change of A into the A and B. And then you have what's called a catalytical process, non-nucleation process, and it's more or less for who are familiar from in nonlinear chemistry is Belousov-Zamotinsky reactions. And later in 1993, another model that was more based on polymerization of actin, tubulin, such kind of things. That was based on nucleation, elongations, pathology. In mathematical words, it's kind of Lipschitz-Yosef continuous, or in Becker-Doring for condensation phase or fast transitions. So nucleation elongations. And in this, you don't have a real catalysis, in apparent catalysis. You need only have nucleations, some bad proteins that come and initiate the conversions too. So the prion paradigm by itself that has been proposed in, let's say, 1997 by Prusiner, that got the Nobel Prize for this, can be decomposed in let's say, three phases. The first phase is that you have the bad proteins, the protein that is misfolded, that come and interact with the good proteins, PRPC, and induce is conformational change, and you have an elongations. Because PRPSC, this aggregate is kind of fibers, so. And you have elongations from this extremity, and you have, okay, the fibers that elongate. But it's not sufficient to describe the pathology, because you have one fibers and you generate only one extremity. So you don't have the amplification phase. So to introduce the amplification concept, the biologists, the biochemists that work on prion said, okay, let's suppose that you can have fragmentations. You elongate, you break these fibers in two, so you have apparition of two extremity at least. So templating, amplification of interface by fragmentations, and then you can have some exponential growth of the number of extremity and the amplifications. And then you have also the spreading through the tissue, and you can have a kind of cycle, templating, amplification, and spreading through the tissue. So this is the fundamental base of prion paradigm. But you're gonna see that's not so simple, and it's more or less a little bit much more complex. A few words about the good proteins and the bad proteins. So I will not go in detail on this because it's out of the purpose of this talk. So this is the structure of the good proteins. This is that protein that everybody have in their brain or their whole body. This is a very simple protein, nothing very special. It's attached on the cell surface membranes, and it's participate to a lot of biological activities in the brain. Now, for the bad proteins, the one that's it's pathogenic, it's infectious. It's a fibrillar proteins. Here you have an overview, and you're gonna see a lot of images later. It's what we call the amyloid fibers, and the structure has been established very recently. I mean, recently, 2021, by a cryo-EM approach that get rise to the resolution of the structures at the molecular at atomic resolutions. So you have a twisted fibers, and this is the interface on which you have the good proteins that stack, and it's converted. Something important, but a little bit difficult to integrate when we are not in prion field it's what we call prion strain. If you look, for example, conventional pathology like flu or COVID, you have different strain. One strain make that you are sick like hell. Another one, you just lick it on your nose and everything's okay. But the only things that change is what we call the strain of the pathogen. But in prion, we have exactly the same. We have what we call the strain, and the strain is encoded in the structure of the assemblies. It's not something that change in the sequence of amino acid, but in the structure of the assemblies. Here, typically, you have two types of strain that are common in human, sporadic CGD and VCGD. One make that you have a lot of plaques in your brain, very lot of plaques, and use a lot of sponges in your brain. The other one, very low amount of plaques, almost not sponges, but kill you much more faster. Huh? And don't have induced degenerations in the same brain area between these two strains. So the strains address what gonna be this the time of how to say incubation time, histopathological lesions, and the incubation periods, and also the brain area that's affected, of course. And this information is only encoded in the structure of the strains. Here, to represent, you have 4 different prion strains, and you see the only thing that's changed is only the conformation of the interface of the replications. Just for giving an overview, for example, in sheep, you have more than 20 prion strains, and the strains is only different in the conformations. So this I can pass. So just to set up what's the state of the art and what we're gonna go further in this talk. Let's say, so in Prion's disease, what's for the moment that is well established and less well established is the anatomical set of prion replication. We know that everything's occur in the brain, where PRPC proteins is expressed. The subcellular part where the prion replications occur is the cell surface, mostly. The mechanism of neurotoxicity, nobody know how the neurotoxicities occur and what's make that the neuron dies. And it's exactly the same as in Alzheimer's or Parkinson. Nobody know how it works. And the structure of PRPSC, and as I told you, has been resolved only recently, and it's gonna bring a lot of things to make good rationale now. And what's important, the mechanism of prion conversions, we have only the model of Griffiths in say 67 and the one at 93 that I show you. And for the moment, the dynamic of prion assemblies, I mean the dynamic of these fibers and other type of assemblies that I will go further later, is for the moment something very open, still an open questions, and it's a very new thematics. Let's say it has more than 6 or 7 years, and the mechanism of prion amplification and spreading to, it's also a large open questions. So mostly in this talk, in my lab, in my team, we mostly work on these 2 group of questions, and today I'm gonna only focus on these two items. So replication, source of the structural diversification. This point is very important because, as I told you, the current dogma make that the replications occur through the templating. So you have the bad proteins and you have the good proteins. Good proteins are going to stick on the surface, and you have the structural change like something like this. And when you look the kinetics of this kind of things, it's something very linear. I mean, you can do whatever you want. You will see only something very concentration dependent, saturable if you are not enough substrate, and it's a single order kinetics. Okay. In the team, in the group, we are able to purify and extract the bad proteins from the brain. We have the good proteins also, and we use some optical approach to investigate the conformational change, the oligomerization, the aggregation of the prion proteins that's based on light scattering measurement. So the light scattering measurement give you something that it is proportional on the second order momentum of the molecular weight of the protein. So it means that when you double the size of the molecular weight of the proteins, because you make 2 times more aggregates in terms of molecular weight, you're gonna multiply by 4 the intensities of the light scattering. And what we did is that we took the prion assemblies, the bad proteins, we put it in the tube, in the cuvette, and we added the good proteins. And what we observed is a first phase of a very rapid increase in terms of size, then a decrease, and apparition of very sustainable oscillations, probably dumping oscillations, and that converge to the very low molecular weight, probably not reaching the initial size, but almost the polymerization. We had more or less the same thing.
Sorry, you meant
Sorry.
very simple question here.
Yeah, tell me.
When there's an increase in size, so what do you measure exactly? Is that the size? Is that the quantity? Is that
No, is the length of the assemblies.
Okay.
It's let's say that the assemblies are a repetition of the same proteins. Okay, what I measure is the square of the I. Is this one?
Okay. Thank you.
Am I clear?
Yeah. Thank you.
Okay, welcome. So yeah, sorry, I represent as this because this, when you multiplied the molecular weight of the proteins, is become the molecular weight square of the size. This slide was done for the biochemist, so I apologize. So we have also the same for another type of prion strains, and for this you have this kind of type of very repeatable spikes, and then probably superimposed of different harmonic of oscillation, and suddenly stop. So I don't have any explanations for the moment of this, but definitively, this kind of things cannot render to account this kind of behavior. Okay, so but in terms of biophysics, and biochemistry, which kind of information we can take from this? That the process of templating is something that is non-linear, so this is quite easy to say. Oscillations indicate that we have a dissipative process, so it means that we have to reach something much more stable, and probably we have apparitions of multiple conformation that exchange between them, in nonlinear exchange between them. Okay, and of course this is incompatible with an interrogation process. This I will skip because it's gonna be something very difficult to introduce you, but you have to take to account that we demonstrate that the prion assemblies, the aggregates that we purified from the brain, in fact, when you just dilute them, they are changing in terms of size. And this is something that was quite real. I will not extend on this. They are super spread dynamics in terms of size distribution. So to go a little bit deep to understand what's this the phenomenon of, as I told you, nonlinear conversions, structural diversification, because you have to have different conformation that compete or exchange to have this kind of oscillations. What we did is that we do what we call in vivo experiments. What does it mean, in vivo experiments? It means that we take the bat proteins, so the prion, we infected in the brain of mice, several mice. And what we did is that we sacrificed at different time the different mice. We looked the size distribution of the prion assemblies by a technique that's called sedimentation velocity that allowed you to separate the proteins according to their size, their molecular weight. So more aggregates, more large aggregates you have, more you go to this side of the gradient. When you have small, you stay here. Okay. So at early stage of the conversions, the infections, what we have is very small assemblies. And more you go in the term of the until the end stage of the disease, more you will have larger assemblies. And when you do this kind of experiments, but in vitro, in terms to have a much more precise size distributions, you have more or less the same thing. But what you observe is that you have small assemblies. First, at the first stage, you have very small assemblies, and these small assemblies disappear progressively, and you have larger assemblies that appear, but in bimodal way. You don't have a translations of the small assemblies into the large assemblies. It means that you don't have small assemblies that progressively go and form large assemblies. You have one that decrease, and you have large assemblies that appear. So this was, I mean, we were very surprised to see such kind of behavior. We are expecting to have a translation from small to large assemblies, and to determine if these assemblies is the same than this one, but only change in terms of size. We purify them, and we do some biochemical and biological characterization that I will not extend on this subject. And we demonstrate that these two assemblies are structurally different. A, what we call PRP-SC A and B, are structurally different. Small assemblies, A, B, very large assemblies. Okay? So we do a lot of kinetical studies, how A are transformed to B, and we put all these together. And in collaboration with Marie Doumic and Mathieu at this stage, we proposed some kinetical schemes that show that you have A that it's formed. We call it A1 here. A is formed at the beginning of the conversions. A interacts with some B assemblies that were present in the initial state, and you have through an autocatalysis B that start to become larger and larger, and go through a autocatalytic process and increase in term of size. But what we so here you have this is the kinetical schemes. This is the experimental observations, and here's the simulations that that we did to render to account based on these kinetical schemes. And okay, it was not exceptional fitting process, but at least we were able to show this this bimodal behavior. So what was important from this step, and this is for in the prion field, it was a breakthrough. It is that the conversion process, the initial process that occurred during the conversions, is that you put a fibers, and from one fibers you have at least two type of assemblies, and this in a deterministic pathway, not something very stochastically that occurred. The diversification doesn't occur in terms of stochastically, it means not due to the environmental perturbations. So this kinetic scheme was more or less the first part of the studies. Then, when you have two types of assemblies that coexist in the same environment and both are able to replicate, the question that occur is why they don't compete, why they are not in competitions for the same substrate, and how they coexist. So for this, I will go through this because it's going to be a little bit long. So here you have schemes of this kind of the assemblies. So this is the BI assemblies, the large assemblies. And what you see here is atomic force microscopy experiment. You have this small object that are the A assemblies. So what we did is that we said, okay, let's say to explore how these two assemblies can coexist together if they are able to exchange the exchange. So what we did, again, light scattering measurement, and we incubate them in the same media. And what we observe, we look just the relaxations, how they evolve as a function of time. And what we observe that when we put these assemblies, first we have a very slow phase of depolymerizations, the size decrease, a catastrophic depolymerization, and occurrence of very sustainable oscillations. Okay, and I think that Mathieu will much more focus on this part. We did this more or less the same thing with other type of prion strains. And what we observed, again, oscillation, perhaps not as much clear as the other one, but also oscillations, sudden depolymerizations that occur, and such kind of things. We did also other technique perturbational approach by temperature rising, temperature decreasing, and again we see oscillation that the frequency change or damping oscillation depending on the type of oscillations. So what we did, and I will probably make a very long story short. We put all this kinetic data and the experimental observations that we get from the experimental approach together. And by putting this, we propose a new kinetical schemes that Mathieu will detail a little bit more. That integrate the structural diversifications, so the process of the conversions, but also the dynamic exchange of the prion, the different type of assemblies, the kinetical aspect. So we integrate a catalytical depolymerizations from A that induce a depolymerization of B, and the conformational change that make the B assemblies transform to the A assemblies transform to B, and make that this kind of oscillations can occur. So Mathieu will a little bit focus much more on this, and the numerical and the study of these kinetical schemes make that the simulations, the first simulation that Mathieu did during his PhD, make that he was able to render to account this kind of depolymerization and occurrence of these oscillations. Here is the simulations, and here is the typical experiments that we obtain. So to summarize and to finalize this presentation, so we demonstrated that the replication process is interscale generate diversity. The prion assemblies are highly dynamic, and this dynamicity contrasts with the deadpan amyloid fibril structuration that it's something that's very well established in the prion field. It exists a complex process of exchange between different PRP-SC subpopulation that make that we have occurrence of this kind of oscillations or nonlinear kinetics behavior that you saw. And finally, that PRP assembly dynamicity and the exchange between different catalytical exchange that you have different conformations, and especially the fact that you generate small object that opened a very good new field to investigate how the neural invasions and tissue spreading can occur, and how prion could impose some histopathological pattern. And finally, a lot of persons contribute to this project. So my group at INRAE, Mathieu from Maillage that was recruited a few years ago now, Marie Dumic, of course, Clemens Fendler, a lot of other persons that contribute for the biochemical aspect. So thank you very much for your attentions, and now if you have questions, don't hesitate.